Chapter 14 covers DNA structure and DNA replication specifically. The first part of this lecture will just be looking at the DNA structure, and then I have a very good YouTube video that is linked into your textbook and an activity for you to do regarding that video, so it won't be a very long lecture. So the history of DNA goes back to Friedrich Miescher, who is credited with discovering nucleic acids in bandages full of pus that he collected in the late 1800s. He was able to extract a weak acid material, which he didn't call it nucleic acid at the time. He called it nuclein, because he knew it was from the nucleus. But that's really all he knew about it. Of course, we know that DNA makes up the chromosomes that are inside the nucleus of a eukaryotic organism, or the chromosome that is found in a location in the prokaryote called the nucleoid region. In about 1950, late 40s, 1950, a biochemist named Erwin Chargaff chemically studied double-stranded DNA from different sources and was able to figure out that no matter what the source of the DNA was, the amount of nucleotides containing adenine was always approximately equal to the amount of nucleotides containing thymine. And the amount of cytosine was always approximately equal to the amount of guanine. And so what he basically was describing, although he didn't know it at the time, was what we call the complementary base pairing rules of DNA. In 1953, James Watson and Francis Crick, pictured here, working in the United Kingdom, figured out the structure of DNA, but they didn't do any experiments themselves. They used the evidence that they gathered from others to propose a double helix structure for DNA. There were other scientists working on this at the time, but they came up with a structure that was consistent with all the data, and eventually won the Nobel Prize in Physiology or Medicine in 1962. So the structure of DNA, you may recall, DNA is one of the nucleic acids, and nucleic acids are made of nucleotides, and nucleotides contain, let's see if I can get this pen to work, a nitrogenous base, in this case thymine, a pentose sugar, for DNA it's deoxyribose, and a phosphate group. So I've circled one nucleotide. And then that nucleotide will be connected to the next nucleotide by what's called a phosphodiester bond, which would be located fundamentally right there. This bond here. Okay. And so you have a long strand. They only show two nucleotides here, but it continues on. And then there's another strand opposite that is also a strand of nucleotides joined by phosphodiester bonds. And the two strands face each other such that the nitrogenous bases of one strand form hydrogen bonds with the nitrogenous bases of the opposite strand. And when Watson and Crick figured this out, what they realized is that if you break apart these hydrogen bonds that run through the middle here, that each strand by itself has enough information for an enzyme to be able to build the missing strand, the complementary strand. And it turns out that is the model of replication that the cell uses. It pulls apart the two strands, the complementary strands of DNA, and then it uses each strand, and because the enzyme knows the complementary base pairing rules, it can put the correct nucleotides in to build a new complementary strand. And so in 1958, two scientists named Meselson and Stahl, Matthew Meselson and Franklin Stahl, proposed, actually they came up with three proposed models of DNA replication, three hypotheses. We call them the conservative model, the semi-conservative model, and the dispersive model. And using an experimental method involving radioisotopes of nitrogen, they were able to demonstrate that the semi-conservative model is correct. So that's what bacteria use, and it turns out it was later also shown that eukaryotes use the same model of replication. And so that's kind of the history and the structure of DNA. But the main part of this chapter is the step-by-step details of the replication process. And so you end up often with a figure that looks like this, which is obviously very complex and doesn't really show how step-by-step DNA is replicated. And so what I like to do is start with the list of enzymes in order in which function to perform replication. And so all of the enzyme names or protein names are in red, and those are what you would need to memorize. But I have a video that that is linked into your textbook, and what I would like you to do is watch the video and have this list next to you, and watch the video and follow along with the steps and start to put this into perspective, into some understanding of how replication works. Now remember, the general goal of replication is to create dyad chromosomes or replicated chromosomes in preparation for a cell division. So this would be S phase of the cell cycle when the chromosomes are replicated. And so what we're doing here is just looking step by step at how that replication happens. You know, possibly more detail than you wanted to know, but it is going to be on the test. So I'm going to stop this lecture segment here, and you're going to go on and keep this list of terms, list of enzyme names, and then watch the YouTube video. And then I'll have some questions afterwards for you to complete.